So here we are talking about evolution and COVID-19. Mutation, selection and COVID-19 variants. Let's see what is the relationship between these terms. Mutation, as we know, is a change in the genetic composition of an organism or even a virus like the SARS-CoV-2. Now this change can be in the sequence of the gene, the structure or number of the chromosome. For example, say this is a gene sequence composed of repeating units or nucleotides ATGC in a given order. A change in certain nucleotides will result in mutation and formation of a mutant sequence. So what is selection? A selection is a genetic change getting its frequency altered and fixed in the population. Say for example, this is a population with certain individuals having a given genetic sequence, which we call as the wild type sequence. And if a new mutant sequence comes into play, and after several generations, that mutant sequence gets altered in its frequency and that alteration gets fixed, we call it as a selection. So how does this mutation occur? Say this is a given genetic sequence that we call as the wild type sequence. Now, during the process of replication of this sequence in the parent organism, by replicating machineries which is erroneous, a sequence with certain errors come into play. However, this is not the sequence that is expected after the end of the replication phase, because additional steps of proofreading is also involved. However, that proofreading in itself is flawed, and hence the final mutant sequence that we get contains certain anomalies in terms of nucleotide. Now, if you compare the mutant sequence with the wild type sequence, we will see that there is a change in a given nucleotide, and this change results in a change of codon. So what is a codon? It is a 3-nucleotide code designating a given amino acid within the protein that is formed from this genetic sequence. Now, this amino acid substitution that we can see here will result in a change from arginine, a given amino acid in the wild type protein, to proline, the mutant amino acid substitution in the resultant mutant protein. Now, what is the response of mutation in the population? Now, selection will give the answer to this question. We have wild type genetic component and we have a mutant component, and this forms a population of organism or even SARS-CoV-2 like virus. Now, due to the origin of this mutation, certain response can be seen. One of the possibilities is that the mutant gene product is biologically advantageous to the offspring, say the offspring viruses containing that particular mutant gene. In that case, we will expect positive selection, and hence the frequency of the organisms or the virus containing that gene will go up in the population. Another possibility is that the mutant gene product is biologically disadvantageous. In this case, we will expect a negative selection, and the frequency of the organisms or the virus particles containing this mutant gene will go down in the population. A third possibility is that the mutant gene product has no biological effect on the overall physiology or biology of the offspring, say an organism or the virus. In that case, we will expect neutral fixation, that is no natural selection coming into play. In that case, the frequency of the offspring containing this mutant gene will be random in the population. Now, COVID-19 variants and key mutations. So this is the SARS-CoV-2 virus causing the COVID-19 pandemic, and the 3 major structural proteins found on the surface are the E protein or the envelope, the S protein or the spike protein, and the M protein or membrane protein. Now, this S protein or spike protein helps in the entry of the virus into our respiratory epithelial cells. Now, the world has known 2 variants, the Delta and the Omicron. These Delta and Omicron are WHO levels. The pangolineage levels for these strains are B1617.2 or the AY lineages, and the next strain clade is 21A for Delta. For Omicron, we can see the pangolineage and the next strain clade as well. Now, if you look at the key mutations of the spike protein, some are given here. There are lot of more other mutations as well. We can see mutations like T95I, G142D. So what are these mutations? For T95I, it means that at the 95th position, amino acid position of the S protein or spike protein, there is a substitution of amino acid from threonine to isoleucine. Similarly, say for example L452R, it is at the 452nd amino acid position that there is a substitution from leucine to arginine, that is from L to R. Now if you look at the two strains, we can see that certain mutations are common, like the T95I. These common mutations help us to understand the point of common ancestry of these two strains, that is the point from where these two strains diverged. We know that currently in a given population there are non-infected or uninfected people, and there are infected people as well who are asymptomatic as well as symptomatic. These two types of individuals form a population. The best way to protect the spread of this given virus or variants of this given virus is that to cut the chain of transmission, because it will reduce the probability of origin of new variants. The more we give the chance to propagate in different individuals, the more is the probability of origin of new variants. So thank you and stay safe.